Hello, everyone. Hello, everyone. My name is Chaitanya Chintaluri. I'm your host for today's Neurotheory seminar series. And it is my great pleasure to introduce our speaker for today's talk, Professor Ina Slutsky from Tel Aviv University. And I would like to introduce her. So Professor Ina Slutsky obtained her PhD degree in neuroscience from Hebrew University of Israel, after which she did her postdoc in MIT in USA, after which she again returned back to Israel, where she's been a group leader in the Sackler Faculty of Medicine at the Tel Aviv University. Professor Slutsky's research focus is quite astounding, and she focuses on synapses, plasticity, and stability in neural circuits. And one of her specializations is to apply these ideas in, for example, in brain disorders like in epilepsy and in Alzheimer's. And for her expertise, she's been awarded numerous prizes. For example, MetLife Foundation Award for her research in Alzheimer's. And in her lab, she uses various technologies, cutting-edge technologies, and scientific tools to explore the brain. Just to name a few, for example, is starting at the molecular level, like with the FRET imaging, all the way to calcium imaging in behaving animals. And all of this is extremely fascinating to me, and I hope you're all as excited as me in welcoming Professor Ina Slutsky for her talk today, which is titled Firing Homeostasis in Neural Circuits from Basic Principles to Malfunctions. And before I hand over to Professor Ina Slutsky, I would like to just mention briefly about the question asking features available in Chromecast, the platform that we're using today. As you can see at the bottom right, in the bottom right of the screen, you would see Ask a Question button where you can ask your question. And the default mode is to invite you on screen, and we will try to get you to ask the question yourself. In case you do not want this, please mention it. I would also like to mention quickly that after the talk, we will have a wrap-up session with the speaker where you can come and speak to Professor Ina Slutsky and the rest of us in a more casual way. This is going to be a Zoom meeting, and we'll send the link much later. So without any further ado, over to you, Professor Ina Slutsky.
Thank you so much, Chaitanya and the team for invitation and for such a nice introduction. And just let me know if you see now my screen.
Yes.
Do you see? Yes, because I see just my screen. I don't see anything else. I apologize. So I will try to imagine all of you, and I would be so happy to see you. So first of all, I missed my invitation to Oxford. Then I missed the invitation to Vienna. So let's hope next year we will meet some nice place, and I will be very happy also to see all of you in Israel. But now we will communicate this way, and you can ask anytime any question you want, and if something is unclear, we can go after my talk or also discuss some details. So today I would like to present you as a major topic of my lab during last, let's say, 5 years, and the major questions that we are focusing on is we are trying to understand the balance between stability and plasticity in hippocampal circuits. Obviously, hippocampal circuits don't need introduction, but I would like just to mention few very important features of these circuits which make it a bit unique comparing to others. For example, when we talk about plasticity, you know that hippocampal plasticity is available and it's very high during our adulthood life and even during aging, right? So only deteriorating in neurodegenerative disease. So when we compare it to, let's say, primary sensory cortex, hippocampal plasticity is really has a critical period. And during last 60 years, as you, or most of you know, of course, we learn a lot about hippocampal plasticity. So about changes in synaptic connectivity, about changes in synaptic strengths, about changes in the intrinsic excitability of neurons. And another feature that I would like just to mention in the hippocampus is the existence of adult neurogenesis. So maybe it's a bit controversial in human these days, but at least in animal models or in mice and in rats, it's existing. So very strong and high levels of synaptic plasticity and the appearance of new neurons, all of these changes can really challenge the homeostatic system and stabilization of hippocampal circuits. And that's what we are really trying to understand, how these circuits change on one side and how they maintain stable on the other side, and how this balance can be perturbed in different brain disorders. And we mainly focus on early stages of Alzheimer's disease and epilepsy. So basically, as I told already, during last 60 years, we learn a lot about experience-dependent changes in hippocampal circuits. We learn about long-term potentiation as a cellular substrate of long-term memory, obviously also long-term depression of synapses. We also know a lot about changes in intrinsic excitability during different types of learning and memory paradigms. And of course, we believe that all of many changes are converted to changes in structure, right? And we know about structural plasticity of spines. And when I asked myself why so many laboratories during so many years devoted all their time to study all these distinct types of synaptic plasticity and neuronal plasticity, the question is that most of us believe, right, that these changes really affect the properties of neural networks. But what we really would like to address, and what I would like to discuss today, is which properties of neural networks are changed and which properties of neural networks remain stable. And if they are not remain stable, we face very unpleasant brain disorders. So basically, what are the basic principles underlying stability of central mammalian neural circuits? The answer to this question is really not complete. I would like just to state that we think about stability of activity in neural circuits, and specifically in central neural circuits, based on control theory coming from engineering. So it's a really abstract form of thinking that we can imagine that there is some parameters that are regulated by homeostatic system, which are maintained at the level or within the range of set point level, and that we have some sensors that we really don't know exactly what are these sensors that can detect deviation from the set point of activity. And then there is a series of effectors that really perform this negative feedback mechanisms that return the level to the set point. So there are a few basic questions that we trying to understand. First, of course, what are the basic neuronal functions that undergo homeostatic regulation? It's a very important question to answer. And the second one, what is the spatial scale of this regulation? So I would like to start from a few ideas that the field generated about the variables or regulated variables in neural circuits by homeostatic system. So today we mainly will talk about firing rates of neural circuits, and I will show you studies that support that mean firing rate or firing rate distributions are homeostatically regulated in different circuits. Firing synchrony or firing pattern can be also homeostatically regulated, but we don't have too much studies on this. There are some ideas that the balance of excitation to inhibition can be also homeostatically regulated at different spatial scales, at the single neuron level, at the dendritic level, and so on. And this balance, as you know, is perturbed in many disorders, for example, neurodevelopmental disorders such as autism. And also some ideas around about really local homeostatic rules about preserving total synaptic strength at the level of individual dendritic branches. For example, we can have one branch with high number of weak synapses versus another branch with small number of strong synapses, and both of them may have similar level of synaptic strength. So we have different idea about variables, and we have different ideas about spatial scale. And obviously, in a very complex situation, we can imagine that different set points may exist with different sensors and with different effectors. But we will start from some more simple case, trying to understand what's going on. So a few years ago, it's almost 5 years ago, my lab started to think how to address this question, because many tools that we had before are not suitable for this type of research, for one simple reason, that homeostatic processes are very, very slow. So if you want to maintain or to follow the same neuron or the same dendritic branch during several days, we need a technique that we can identify the same neurons and we can follow them during days and many, many hours. So obviously, hippocampal slices are not useful for this. And we have only two alternatives, one ex vivo cultures and another alternative, obviously, in vivo behaving mice. So we started to address this question from ex vivo hippocampal cultures. And the experiments that we did, in principle, are very simple. So it took time us to establish very long-term recordings. So in this case, we use multi-electrode array. So today we record from 120 channels in 4 amplifiers in parallel, and we can add drugs, or we can add genetic perturbations, or we can add stimulation. And in this case, the examples that I show you that we add baclofen, which is agonist of GABA-B receptors. We choose it only for one simple reason, because GABA-B receptors are expressed almost everywhere in the brain, and the inhibition is very, very pronounced in both excitatory and inhibitory neurons. And you can see y-axis firing rate per unit, that units become almost silent after 10 micromolar baclofen concentration. But in the presence of this perturbation, you can see gradual return of firing rates to the same level it had before the perturbation. If we don't perturb the neurons, you can see that firing rates are pretty stable and nothing interesting happened. So when we average many, this was an example of individual experiments. When we average many experiments together, you can see that following inhibition induced by baclofen, the network mean firing rate comes back to the, let's say, set point level it has before, almost around 1.5 hertz. And in contrast, control cultures, not perturbed cultures, had very stable mean firing rate. So when we looked at the distribution of firing rates, log normal distribution, similarly as has been shown by Yuri Buzaki lab in vivo, there is no statistically significant difference between baseline firing and firing 2 days in the presence of baclofen. So we can say, based on these ex vivo experiments, which are performed under very controlled environment, there are no different structures that connected to hippocampus like in vivo. There are no behavioral states. There are no arousal states. There is nothing like this. So under very, very controlled environment, we got homeostatically preserved mean firing rate and firing rate distributions at the population level. So I hope you can agree with me on that. So just to show you how the data looks like, obviously it's after spike sorting. This is the firing rate in cultures, just baseline firing rate. So you can see synchronized and unsynchronized activity. This is firing rate in the beginning of baclofen, 4 hours after baclofen addition. And you can see it's very easy to detect reduction in mean firing rate and increase in the synchrony of firing rate. And if we look the end of the experiment, 2 days in the presence of baclofen, I hope you can detect that network firing rate came back, and the pattern also in the same paper we show came back as well. So one important emphasis that I would like to make here, that if we look on the network level, as I showed you until now, we can conclude, right, that before application of an baclofen and 2 days in the presence of baclofen, we have almost indistinguishable, very similar network dynamics. But let's now look at closer in the microscope at the level of synapses, yes, and at the level of individual neurons, what's going on. And I would like to show you that each parameter that we quantified, and we did not quantify everything, but some of parameters have been changed. For example, when we looked on release probability of synaptic vesicles, okay, we can see that 4 hours after baclofen and 2 days after baclofen, there is an increase in release probability, which is served in order to bring firing rate back, okay, to the set point level. If we look at miniature excitatory postsynaptic current, again, both the amplitude and the frequency of miniature EPSC are increased 4 hours and 2 days in the presence of baclofen, meaning that synapses have more AMPA receptors and they have more efficient release of glutamate. And finally, if we look at intrinsic excitability of neurons, so when I say intrinsic excitability, that means that we measured it in the presence of synaptic blockers, you can see that the neurons become more sensitive to the same input. So to the same current injected, neurons now fire more, and also membrane potential become more depolarized. So you can see that if you remember now the previous figure, this one, right? When we saw that the distributions of firing rates are similar before and after application of baclofen, but all the parameters, okay, have been affected. So again, as the very beautiful computational work of Yves Marder predicted, that now we have a network that composed completely from different components. And still at the population level, we have the same phenotype of firing rate distributions. Okay. Another important question that we wanted to address at that point is whether other parameters are also homeostatically preserved. And obviously, we wondered what's going on in terms of synaptic plasticity. And when we quantified short-term synaptic plasticity using imaging FM dyes and the same data we got for double patch recording for electrophysiological excitatory to excitatory neurons connections, short-term synaptic plasticity has been really impaired. So you can see that facilitation, which is normally characterizing CA3, CA1 hippocampal synapses, has been reduced after 4 hours and disappeared completely after 2 days in the presence of baclofen, meaning that impairments of short-term synaptic plasticity may be a trade-off, like resulting from systems' effort to maintain firing stability. And the biggest question really is that we would like to understand in connection of to design of network and in connection to brain disorders is how to compose the system from which components in order to preserve stability and improve or preserve synaptic plasticity. Yeah, so this is really important question to answer in future. So now let's see what's going on in vivo. So I showed you our results again. The results are robust, but all of these results came from ex vivo culture, very simple cultural network, right? That have recurrent activity and don't have inputs. So majority of data that we have today came from V1 visual cortex in vivo. So very beautiful data come from laboratory of Gina Turrigiana. All the work has been performed during critical period of plasticity when one eye has been closed, monocular deprivation, classical paradigm, and the firing rates in deprived hemisphere have been measured before monocular deprivation and during monocular deprivation using single unit recordings again. And you can see that firing rate after 1.5 days or 2 days was significantly reduced. And during next few days, they came back to very similar level to the baseline. So here is quantification following monocular deprivation and during monocular deprivation, firing rates dropped and returned back to the set point level. And I would like to emphasize that these recordings have been done in monocular V1. And after eye reopening, this is a new paper just recently published in Neuron, you can see that firing rates go up and again return back to the same level as they were before eye reopening. Another series of studies, very beautiful studies, have been done by Tara Keck lab in UCL. And in this case, all the experiments have been performed by calcium imaging into photon microscopy and in adult mice. And it has been shown that excitatory neurons on average show, following sensory deprivation, show drop again in activity, and activity came back. Okay, again, on average, some neurons did not come back to the set point level. However, inhibitory neurons that have been classified by kinetics of GCAM6 transients, inhibitory neurons have were not, their firing rate was not really homeostatically controlled, and their firing remained much lower than it was before. So this is different in comparison to studies of Gina Turrigiana lab, who showed that fast spiking neurons and regular spiking neurons return to the set point level. So it could be methodological difference or developmental difference, critical period versus adult mice, but that's what we have today for in vivo data. And obviously it will be very interesting to see how different circuits with different functions behave and homeostatically maintain or not their firing rates. So the second important question that we would like to know the answer to it is, do single neurons really maintain their own firing rates, or firing rates are more preserved at the level of networks? Again, from V1 visual cortex, from the same work of Hengen from Turrigiana lab, you can see that following monocular deprivation, there is a drop in mean firing rate, which is returned during monocular deprivation, and few small fraction of neurons have been recorded and recognized during long-term 9 days of recording. So these neurons preserved their waveform. These are extracellular recordings. And as you can see, in this case, the return was really precise to the set point level at the single neuron level. Later study showed similar data for eye reopening. So following eye reopening, again, neurons, some fraction of neurons with similar waveform of spike came back at very precisely to the same level of firing that was before eye reopening. So basically, these studies of Gina Torrigiano lab suggest that firing rate can be regulated at the cell autonomous level in monocular V1 cortex. So the question of what happens to a large population of neurons, right? And what happens also to different type of circuits. I would like just to show you that in our work in hippocampal cultures, the results were different. So, big advantage of culture that the neurons in culture, in mature culture, don't move. So we could record from the same electrodes, the same neurons during the many days. And in this case, you can see that even without any perturbation, so look here, baseline mean firing rate, and 2 days later, the same baseline mean firing rate, you can see this is log-log plot that there are very big changes, right, in firing rate of the same neurons, and only green neurons remain stable. So the green is based on bootstrapping statistics. And after addition of baclofen, you can see that basically it's the same picture. So here we had 80% of neurons which didn't preserve mean firing rate. And here we have even more, 85% of neurons, but basically majority of neurons don't preserve their mean firing rate at long time scales. So we concluded at that time that firing rate is unstable at the level of single neurons, and firing rate compensation is not precise at the single neuron level. So I would like just to bring your attention that the correlation is very nice, but it's not precise, and precision is the key for homeostatic system. Obviously, the huge questions that you, of course, answer yourself, I'm sure, what's going on in vivo. And this is really data that we just now analyzing, and I'm showing you our first initial analysis that in C1 of behaving mice, so here we used calcium imaging by microendoscopy and mice just moving around in open field, you can see that even mean firing rate with difference of 1 day, and the same we have for 2 days, 3 days, and 4 days difference, is very, very variable in C1. And here we are looking only in excitatory neurons since GCAM6 is expressed under CAMK2 promoter. So you can see again the small fraction of neurons remains stable, and majority of neurons, and here we have really large scale recordings around 1,000 neurons per mouse, that majority of neurons are unstable. I'm speaking about activity, of course, right? Are unstable in behaving mice. And currently we're analyzing the same data following perturbation. So hopefully next time I can show you this data. So again, I would like just to conclude that we need, right, to sample more circuits and to address this question whether different circuits with different functions have similar or different homeostatic rules, and this is really important and interesting question to address in future. So another thing that I would like to emphasize is that firing rate set point seems to be regulated by behavioral states. So if we look existing data in distinct cortical circuits, we can see, for example, here is a classical paper by Vlad Vyazovsky, Chiara Cirelli, and Giulio Tononi that shows that in barrel cortex, there is very strong reduction and robust reduction during non-REM sleep. Okay, so if we compare wake state and non-REM sleep, there is very significant difference in mean firing rate. And if we look at different states, we can see that again across network, the non-REM is characterized by low mean firing rate than wake state, and there is also difference as function of sleep pressure in the same state. The laboratory of Yuri Buzaki showed that not only mean firing rate can be affected, but also the distribution of firing rates can be affected. For example, in his recordings in different parts of cortex as prefrontal cortex, cingulate cortex, motor cortex, and others, you can see that in non-REM state, high firing rate neurons reduce their firing, and distribution of firing rates become more narrow and more homogeneous. In contrast, in REM states, there is a reduction in mean firing rate, and the whole distribution moves left. Very similar data has been obtained in the hippocampus, in CA1 hippocampus by Diba lab. His lab really nicely showed that, again, during non-REM state, pyramidal neurons have reduced firing rate comparing to active wake state, and a similar tendency has been shown for inhibitory neurons as well. So basically, our own recordings support regulation of firing rate set points by arousal states. So when we did calcium imaging in parallel to LFP and EMG recordings in the same mice and imaged CA1 activity during wake dense states versus sleep dense states, you can see here's a raster plot that in sleep dense, and it's mainly non-REM sleep, there is much less neurons which are active. And when we are looking at the distribution of firing, you can see that the total level of activity is significantly reduced during non-REM sleep in wild-type mice. However, again, I would like to emphasize that situation could be different in different parts of the brain. And again, V1 visual cortex looks like to be a bit different. For example, a recent study from Yuri Buzaki lab showed that in V1 cortex, the regulation by arousal states depends on the layer, and mainly deep cortical layers 5 and 6 show pronounced change between wake and non-REM states, but layer 2, for example, 3 and 4 don't show any change. And Gina Torojana's work did not find any change in mean firing rates as function of states in monocular V1 cortex. So they recorded through all the layers, and in this case, they did not find any dependency on the behavioral states. So again, the conclusion that different circuits can behave differently, and again, I think large-scale recordings are really needed here in order to understand how the population and large population of neurons behave in different parts of the brain. So the next questions that I would like to touch are, what are the core molecular mechanisms or the core molecular machinery that underline this firing rate set point regulation, whether firing rate set points are dysregulated in early stages of Alzheimer's disease. And I would like also to address whether this homeostatic dysregulation of firing rates is state dependent. So we will start from the mechanisms of set point regulation. And we suggest a dual challenge approach that in principle can really identify the core homeostatic machinery. So what this approach means? First of all, we suggest that most of neuromodulators or activity modulators, they induce a typical homeostatic response or what we call type 1 perturbation. So it means that if we inactivate or hyperactivate network has a capability to come back to the set point level. Okay, so something that we discussed before. However, some molecules that belong to core homeostatic machinery, or what we call type 2 perturbation, can alter set points or can impair a homeostatic response. In this case, we can identify them by looking on changes in basal firing rate, such as, for example, for set point regulators. In this case, they change stably basal firing rate, but do not affect response to type 1 perturbation, okay? But in the second type, we can find molecules that don't affect basal firing rate, but they really perturb homeostatic response to type 1 perturbation. And these are the cases that we are looking for, and how we can identify them, how we can identify the molecular mechanisms that is responsible for different nodes in this homeostatic system. So basically, we thought that different modules can be responsible, can be part of this core homeostatic machinery: calcium, immune, energy homeostasis, proteome homeostasis, genomic stability. And today I will just show you one of the examples that we identified that relates to this energy homeostasis model. And we decided to start this journey from unbiased computational approach based on genomic scale metabolic modeling. And we asked which metabolic genes, okay, so we focused specifically on metabolism here, can bring metabolism of unhealthy, and in this case, we worked with epilepsy patients and epileptic mouse and rat models. So which genes can bring unhealthy metabolic state of network to the healthy one? We just knock out individual, in silico, of course, individual genes from 1,500 genes. The best candidate was DHEDH. This work was published. So this is dehydroortat dehydrogenase that belongs to the molecules which are localized in mitochondria and participate in two different types of reaction. One is de novo pyrimidine synthesis. And the second one is electron transport chain reaction in the mitochondria. So we never heard about this molecule in context of neurophysiology, but we knew that this one is very important for development of neural system. So then we asked what DHEDH inhibition by specific inhibitors that we could just find. The name is teriflunomide. How does it affect the mitochondria? And the results were very striking. We found that inhibition of DHEDH increase calcium transients in the mitochondria during stimulation by burst of action potentials. So you can see that following inhibition of DHEDH, there is stronger buffering of calcium from cytosol to mitochondria. However, opposite situation was occurred for resting mitochondrial calcium. So inhibition of DHEDH significantly reduced resting calcium in mitochondria, and this could be washed out by the drug. So obviously the next question was, how does it affect the function of the system? We started from slices. You can see that CA3, CA1 synaptic transmission was significantly inhibited, input-output is reduced, and short-term synaptic plasticity was enhanced. But then, of course, we wondered how chronic inhibition of DHEDH affect different components of the system. And anything always we measured has been inhibited. So mini EPSC amplitude reduced, the frequency is decreased, maximal firing rate, this is intrinsic excitability measurement, has been significantly reduced. However, very interestingly, maximal firing rate of fast spiking interneurons has been increased. In this case, it's PV positive interneurons. And when we looked at the balance of excitation to inhibition, in this case, this is spontaneous iPSC and spontaneous EPSC measurements in the same neuron. Just by playing with reversal potential, you can see that E-I ratio is significantly reduced due to significant reduction in excitatory conductance. And finally, when we asked how the network firing rate is affected, we found that inhibition of DHEDH stably and persistently reduced mean firing rate, so there is no compensation, as you remember, for type 1 perturbation. But if we add now type 1 perturbation, like a baclofen, you can see that system did not disable capacity to compensate, but all this compensation came to a new set point level established by DHEDH inhibition. So important to mention that this was extremely robust between experiments. So you can see that in each individual experiment, basal mean firing rate is different, could be 5 Hertz and could be 2 Hertz. However, DHEDH inhibited mean firing rate, and after baclofen, it came back to the same level of inhibition per individual network. And this new set point was also very stable. So when we recorded during many hours following application of baclofen, you can see that this new set point is stable and not coming back or not reducing anymore. So finally, we asked, and we say if it's a real set point regulator, then we have to see the same effect for hyperactivity. So we increased activity by TBOA, which reduced reuptake of glutamate, and you can see that network is increased network firing and came back to the same set point level. So basically, we reproduced similar results in vivo in behaving mice by ICV injection of teriflunomide, since it does not pass blood-brain barrier. And we showed also that in pharmacoresistant model of epilepsy, like Dravet syndrome, which is a catastrophic childhood epilepsy with mutation in sodium 1.1 channel, teriflunomide reduced ventricular spikes and decreased temperature threshold for seizure induction. So just to summarize this part, I would like to say that I would like to emphasize the power of genome-wide computational approach to predict homeostatic regulators, and that we found that mitochondrial DHEDH plays a central role in this firing rate set point regulation, that firing rate set point are adjustable, that compensatory mechanisms and set points are separately regulated, and that lowering firing rate set point may be a new strategy to treat intractable epilepsy. I just would like to say that after we published this work, we found a metabolic study by Chiara Cirelli lab. In this study, they showed that orotat and uridine were much higher during wake states than during non-REM sleep states. And when they measured these orotat and uridine levels as function of slow wave activity during sleep, you can see the higher the sleep pressure or the higher the slow wave power, the lower the concentration of orotat and uridine. And I just would like to mention again that orotat is direct product of DHEDH. So it's a product which is converted from dehydroorotat by DHEDH to orotat, and uridine is the last step of de novo pyrimidine synthesis pathway. So based on these results, we predict, and we now make in transgenic mouse to check our prediction that sleep may inhibit firing rates in the end via DHEDH, and this is very interesting prediction, and will be very interesting to see how DHEDH affect the sleep-wake states themselves and firing rate regulation during these states. So the finally, I would like to address. Uh,
Before you go ahead, maybe you can answer one question.
sure.
Uh,
I just don't see
so,
anything. I'm sorry. I see only my
no,
screen.
that's okay. I'll read the question to you.
Hmm.
So, Nico asks homeostasis requires a precise mechanism to return to baseline after a perturbation. Is it plausible that this return is simply a consequence of activity and dynamics rather than having a dedicated mechanism? For example, 2 days after baclofen equals to release of inhibition.
But releases of inhibition is still an active mechanism. So I a bit not really understand. I want to say that this active mechanism, and this was the point of slide, suggesting that there are many different ways to come back to a set point level, but most of them are active mechanisms.
Okay.
So maybe I misunderstood.
We can go back to this afterwards.
Okay,
The
sure,
person still wants. Yeah.
sure. So the last question that I would like to address is whether really firing rate homeostasis fail, maybe in the early stage of Alzheimer's disease. During last 2 years, we wrote a few like perspective and review article about this. And the basic idea came from human data in Alzheimer's patients showing that networks are extremely unstable in 40% of Alzheimer's patients. In this case, patients were in the sleep lab during whole night, and the EEG recordings have been made. And during the whole night activity, 40% of patients showed abnormal, I would say, epileptic-like activity in comparison to 11% of age-matched control. And intracranial recordings in a few Alzheimer's patients show that there are silent seizures, silent seizures in the hippocampus, while nothing have been detected by EEG recording. So this data suggests that some instability occur in the brain and specifically in the hippocampus of Alzheimer's disease patients, and the question why and how. So we decided to address this question using young Alzheimer's disease model mice. These mice have familial Alzheimer's disease mutations, and they have a lot of pathology in their brain. So amyloid beta is abnormal. There is much more long forms of amyloid beta than short forms, meaning that the tendency to aggregate of amyloid beta in the brain is increased. There is more soluble amyloid beta, and there is already a start of plaque formation. So this is an animal model. However, at this young age of 4 or 5 months, still we could not find any change in sleep-wake cycle, and we have much more detailed analysis. So sleep-wake cycle is different around 9 months of age, but not at 4 or 5 months. And we still could not find any changes in hippocampus-dependent memory function. For example, special working memory was normal at this age, and also contextual fear memory was normal at this age. And in order to maintain cognitive function in face of developed pathology, right?
There seems to be some issue with the connectivity. So please wait while we connect back. Yeah, there seems to be an issue, and we fear that Ina might not realize that she's away from the talk. So please hold on while we try to get in touch with her. So if there is anybody who has contact number or some way to reach to Inna, please get in touch so that she knows that her talk is frozen.
Hello?
Okay, now we can hear you again.
You didn't hear me at all?
Sorry, we sort of lost you from the second part that you started with the Alzheimer's, and your screen froze,
Oh.
unfortunately.
Hmm.
Hold on a minute. So maybe you can go back to the slide with the part two. Can you share your
Yes.
screen again, please?
When did you lose me?
I'll just tell you with when you started the second part of your talk, second question, and third slide from there.
This?
I can't see your screen at the moment.
How do I share again? Okay. Share. Okay. Do you see?
Not yet. Yes.
Now?
Now, yes.
So this one, did you see?
This one, you were here when you froze.
Okay. So
Slide.
you want
Yes.
you want me to briefly explain?
Yes, please.
I'm
Thank you.
sorry. Okay. So basically, what we did, we took Alzheimer's disease model mice, which have pathology. Okay, so there is a very clear amyloid beta dyshomeostasis, but at 4-5 months old mice, there is no evident changes in memory or hippocampus dependent memory. So they have normal special working memory, normal contextual fair memory, and normal sleep-wake cycle. So basically, these mice have very active homeostatic system protecting them from failure in memory and in sleep-wake cycle, right? And then we ask what's going on in CA1 neural circuits of these mice. So we used calcium imaging using microendoscopy. Do you hear me? I hope you are. So using,
Yes,
yes, okay, microendoscopy in behaving mice. So we can detect thousands of neurons in CA1. And you can see this is raw data. This is already processed data. This is the raster plot running. And we use the system to really understand what's going on at different stages. So after investing a lot of time in establishing it, and again, we use CAMK2, GCAM6 virus in CA1. So after establishing all of this, we were very disappointed. I will briefly just say what we found, that we could not find any difference in distribution of firing rates. And here are data for 6 mice, around 4,000 neurons, no change at all in distribution of firing rates at active awake state in mice running in open field. So it was really huge disappointed after all efforts invested. And then we say, okay, maybe something happened to synapses. And we recorded shuffle collateral CA3, CA1 input output, and we could not find any difference also at these young mice at this stage. And then we ask, maybe we can look at different states of arousal. And we decided to anesthetize mice because, general anesthesia really causes very low state of arousal, right? Then, first of all, we took wild type mouse, and you can see data in a awake mouse and data in a deeply anesthetized mouse under burst suppression state of anesthesia. And I hope it is very easily for you to see that activity during anesthesia is reduced and become more synchronized. So when we look at raster plots and LFT recordings, you can see a very significant drop in activity already after moderate anesthesia. This is 1% of isoflurane, and even stronger decrease and synchronization under deep or burst suppression level of anesthesia, 1.5% of isoflurane. And you can see very significant, and there is no surprise, it's a feature of general anesthesia, right? Very pronounced inhibition of CA1 activity. And the next question that we ask, if we can find this similar physiological effect of anesthesia in Alzheimer's model mouse. And here the big surprise came. So when you look the data, this is awake mouse, and I just remind you that we could not find any difference in awake state, in active awake state, and this is a deeply anesthetized mouse by the same level of isoflurane. I hope you can find huge difference. So you can see that under anesthesia, there is very strong and synchronized activity in Alzheimer's mouse. And if you look at raster plots, so it's almost very small inhibition, or in some mice we did not see any inhibition during anesthesia of activity level under anesthetic depths, moderate and deep. And if we look at raster plots, you can see that firing rate distribution are right shifted, so there is stronger median calcium rate, and total level of activity were not inhibited as we found in wild type mice. So basically, this result suggests that the activity should be hypersynchronized. And we did this experiment, this is electrophysiology, in 3 different mouse models with 3 different mutations. And we found very robustly that only under anesthesia, both moderate and deep, there is huge difference between wild type and Alzheimer mouse. I don't know how many of you know it, but these are interictal spikes, which are pathological activities that you see in APPPS1 model and in other models as well. So under low arousal states, we exposed, right, the pathophysiology of these mice. And the last slide, of course, last questions that we asked, how these changes occur during sleep. I already showed you that during non-REM sleep in wild-type mouse activity is reduced, but not in Alzheimer model mice. And very important, again, to emphasize that we did not find changes in sleep-wake cycle and changes in slow wave activity. So this is really initial changes in firing rates, firing rate regulation during sleep stages at very early stages in mice. So these are the results. I will not go over all the conclusions again. And I would like to thank my students, because if you did not hear, this is the most important part. So the students are really amazing. And I showed today the work of Daniel Zarhin, who did all the in vivo study in Alzheimer's and in DHDH project, Bo Stier, who invest a lot in conceptualization of all the homeostatic part, and Nir Gunen, who did metabolic modeling for DHDH project, and my collaborators, Yaniv Ziv, Yuval Nir, Dorit Derdikman, and Eitan Rupin for the study I showed today, and funding agency. And thank you so much for listening. I'm really sorry about problem with the lecture.
Okay, thank you very much for your talk. And on behalf of everyone, I clap. And so I have a question with regard to the initial perturbations that you showed with the baclofen. So after you induce this, after you inject this baclofen, even though the rate of firing decreases quite dramatically, it seems to not go down to zero, and there seems to be ever so slight amount of background synchronous firing. Do you know why this happens? Like just
So it's really the data that I showed you was already 4 hours in the presence of baclofen, and it's really concentration dependent. So we can I can show you if this is 10 micromolar baclofen. If I add 1 micromolar baclofen, it will be 50% reduction. If I put 50 micromolar baclofen, it will be 0 firing rate. Okay, but we found that irrespective of, because you can argue, right, that the larger the perturbation, this was the biggest criticism for the field, I think, that everything is physiological and all this homeostatic system, I heard it so many times, are only relevant for pathology, and it's not physiologically relevant at all, right? And we have many different types of perturbation, and you can really calibrate the system, showing that the homeostatic system work for small perturbation as well as for big perturbation, for up and down, okay? So when we are talking about sleep, for example, the differences in firing rate are not so dramatic, right? You can find 30, 40% difference, but these are, again, physiologically relevant differences. So in order to understand it, of course, always scientists try to make a strong perturbation to get big difference in order to understand what's going on and then to fine tune it, right?
Yeah.
But
I understand. So the other question I had was related to this Alzheimer's research that you showed with regard to the human patients. From, from at least from what I know of the Parkinson's research, for example, there seems to be drastic changes in the sleeping patterns of Parkinson's patients much earlier before any of the pathological symptoms arise. Are there any similar kind of things in Alzheimer's? And
so in Alzheimer's, yes, it's a good question. In Alzheimer's, early study suggested that at very late stages of Alzheimer's disease, there are sleep-wake cycle problems. But now more and more data suggesting that it could be also occurring at pretty early stages. I don't know if they precede cognitive decline or not, but they definitely precede very gross neurodegeneration and spine loss and neuronal atrophy. So I did not show this slide, but today we know that pre-symptomatic stage in human Alzheimer patients lasts around 2 decades. Okay. And it is initiated by dyshomeostasis of amyloid beta. This is the first sign of the disease. And the biggest question for us, right, is to understand how we can recognize disease before cognitive dysfunction, because it already, when it happens, it's pretty, everything is pretty lost already. So
okay. Yeah, please. Go on.
Hi, Peter.
Hello, Ina. Very nice to see you. Very nice
to
talk.
see you.
I have a short question. We have time later. Does the baclofen paradigm induce any structural plasticity, or is it purely functional plasticity at synapses and excitability?
It's a very good question. So we just recently got funding from DFG on this topic in collaboration with Alessio Attardo, who is doing structural plasticity in vivo in CA1. And we are going to address these questions. But I am almost, so the initial study of Chris Harris using other approaches, not baclofen, showed that there is structural plasticity at the level of spines already at 8 hours of perturbation. So I'm almost sure that this is a part of the story. Yes.
Thank you. Thank you. Talk to you later.
So the next question is from Anissa. I've invited her to the screen. But maybe I can. Oh, she's here. Hello, Anissa. Okay, I can ask the question on her behalf. So she asks, is there a change in the DHO-DH expression level or activity in APP PSI mice?
So this is something that we are working on this now. So I cannot really say 100% yes. To change to check activity is not simple. So the assays are not very robust. So we think about collaborating with some labs working on this, and hopefully we will get some answer. So obviously it's a good question to ask, and we would like to know the answer.
Okay, if there are no further questions, I think we can end this session. But I invite you all for a wrap-up session, a Zoom meeting with the speaker, and I'm just pasting the link in the chat room here. And you can come and continue the discussion there. So on behalf of everybody, thank you very much, Inna. And I apologize for the technical issues that we had today, but in the end, I think it was a great talk. Thank you very much.
Thank you.
See you in a bit in the Zoom room.
Thank you.